Hey all, I'm Carlos. I'm currently a postdoc at the University of Kentucky, and today I'm going to be talking about some of the research I did on the evolution of monitor lizards while I was at the Australian National University. First of all, I want to thank you all because you're doing way more for science literacy than most of professional scientists, and you are training the next generation of scientists, so thank you for that. Okay, so I'm going to start by telling you a little bit about myself so that I'm not just this anonymous guy talking to you. So I grew up in Mexico. I'm actually sitting in my childhood home right now in Mexico City. And I have always been obsessed with animals. I'm that dinosaur kid that never grew out of it. And then I went and did a bachelor's in biology at the National Autonomous University of Mexico. My research focused on the systematics of the earth snakes from Mexico. This is a very diverse group. There's around 40 species. And I was focusing on a population that was an undescribed species at the time. Then I did a master's in Mexico as well, working on blue-tailed skinks. And I did systematics as well, so looking at one particular species and testing whether it's a single species or in reality it represents more than one species. And we described several new species as well of blue-tailed skinks. Then for my PhD, I went a little bit far away from home. I went to the Australian National University in Canberra. And there I worked on monitor lizard evolution, which is still a major focus of my research and which is what I will be talking about today. Then I did a short postdoc in CUNY in New York City, in particular in CityTech in Brooklyn. And I was working on how new species are originating in the arid regions of North America, focusing on phrynosomatic lizards, which are these really cool lizards, including the horny toads that squirt blood out of their eyes. And currently, as I was saying, I'm a postdoc at the University of Kentucky, and I'm working on the evolution of a particular family of salamanders called the true salamanders and the newts. In my free time, I like to go out into the field with friends and look for animals. And as you can see, we are all obsessed with wildlife. This is a bandy, a snake from Australia. And things can get a little bit awkward when all of us are trying to photograph the same animal. Yes, that is 3 grown adults on their bellies trying to photograph a lizard, which is somewhere around here. And things can also get a little bit awkward if one of your friends decides he wants to try handstands. But yeah, we've seen many cool animals throughout the years. This is a green tree viper from Mexico, southern Mexico. This is the thorny devil from central Australia. These are 3 kangaroos from northern Australia and cassowary from northern Australia as well. Another thing I like to do in my free time is playing soccer. We won one of the intramural tournaments in ANU. And as unlikely as it may sound, I sometimes combine these passions. So this is me submitting my PhD thesis by kicking a ball into a mouse, and I still don't know how that worked. Well, now let's talk a little bit science. So the questions that I'm interested in are actually pretty simple, but they can have really complicated answers. I'm interested in why organisms look the way they do. Like, why does this chameleon have the horns and the weird googly eyes? Why animals live where they do? Like, why do koalas only live in Australia and African elephants only live in Africa? And why some groups are more diverse than others. So, for example, we only have a handful of great apes, including us, humans, but we have thousands and thousands of species of beetles. And we can look at each of these questions at 2 different scales. So let's take the question, why do organisms look the way they do? We can look at it at the ecological scale, like in this study, where they looked at the behavior and appearance of a native and an invasive lizard when they live together. And they found that when they have been living together for a few years, there's some behavioral changes. One of the species starts living in higher up in the trees and the other one lower up in the trees. Whereas when they're separated from each other, they occupy the same height. And this is to avoid competition with each other. And when decades have passed, these behavioral changes get followed by changes in morphology. So one of the species develops shorter limbs and another species develops longer limbs. We can also look at these questions, why organisms look the way they do, at the macroevolutionary scale. Like here, where they compare the morphology of different morphologies, the physical appearance of organisms. And here they compare the morphology of the different cichlid species. These are some fish from Africa, and how the morphology relates to how these fish are capturing their prey. But historically, there's been a disconnection between these two scales. My research is focuses on using large genetic, morphological, and environmental data, as well as newly developed methods to try to link these two scales and discover how ecological processes are shaping macroevolutionary patterns. And now I'm going to introduce you to monitor lizards, which are objectively the coolest animals on Earth. Monitors belong to a group that includes the iconic Komodo dragon here, and around 80 other species that have a wide distribution in Africa, Southern Asia, Australia, and many islands in the Pacific. Most of them are carnivorous, such as this Komodo dragon, and the largest species, such as the Komodo dragon, can overpower large prey, such as buffalo and humans, and it was recently discovered that they can do this with the help of a venomous bite. But there's also a few species that feed on fruit and leaves. And there's some variation in how the different species of monitors look. Here on the left, we have a very thin species that lives up in the trees, and they have very long tails and very long limbs to climb around in the trees. Whereas this terrestrial species over here is a bit fatter, stockier, and it has shorter limbs and a shorter tail. And there's also some variation in the shape of their heads. This is a species that lives in rock crevices, and you can see that it has a very very flat head, so that it can hide between the rock crevices. And this terrestrial species has a very deep head. But perhaps the most striking variation is in body size. This is the smallest species, which is the Dampier Peninsula monitor of Northern Australia, and it can fit in the palm of my hand. Whereas this is the largest species, the Komodo dragon, which is a little over 3 meters long. And to characterize this variation, I visited several natural history museums across the world to measure thousands and thousands of monitors. And this one here is the National Natural History Museum in France. And here I'm measuring a crocodile monitor in the Alexander Koenig Museum in Bonn, Germany. So I recorded all these measurements, such as body length, neck length, head length, and same for the limbs. And I also characterized the variation in head shape for thousands and thousands of specimens. And while physical features are a good indicator of evolutionary history, DNA provides a much more detailed picture. So I took a 390 gene dataset that was generated by a friend in 2021, and I used it to infer the family tree of monitor lizards. So this tree is simply telling us which monitor lizard species are more closely related to each other. And I'm going to be talking about these 4 research projects, which are all focused on ecological processes and how they shape microevolutionary patterns using monitor lizards as a research model. First, I'm going to be talking about the abiotic and biotic drivers of monitor diversification. Then I'm going to be talking about incumbency effects in Australasian monitors. Then I will be talking about the ontogenetic drivers of phenotypic evolution in monitors and their close relatives. And finally, about hybridization in a group of large Australasian monitors. And there may be some words here that you don't recognize, but I will talk you through it as we go. Okay, so hoping because we're going to learn about the ecological drivers of evolution. And for us that don't know, GOANNA is actually the Australian word for monitor lizard. And first, we're going to talk about the abiotic and biotic drivers of monitor diversification. So it is well known that abiotic conditions have an impact on the number of species that conform a particular group. In this study, they found that as the Andes Mountains increase in elevation with time, so here in black, here we have the past and here we have the present, and you can see that the average elevation of the Andes is increasing. And as they increase in elevation, the number of species of this group of lizards increase as well, likely because populations were becoming isolated in mountaintops and they were eventually differentiating to form different species. Abiotic conditions, such as temperature, precipitation, can also influence the physical appearance of organisms. In this study from 2020, they found that rodents' tails are shorter in colder regions, and while they are longer in the tropics, likely to reduce the loss of heat through the tail in the colder climates. Okay, so I was telling you about the importance of abiotic conditions, but what about biotic conditions? The species that one coexists with can theoretically have an impact on evolution. So you've probably heard of adaptive radiation, where one species diversifies over time to give rise to many different species that are each interacting with their environment in a different way. Like here, these birds from Hawaii, from a common ancestor, they diversified into many different species that are feeding on different things. And so they have a different shape in their beaks to eat different things. And it's thought that they do so to avoid competing with each other. So I use the evolutionary tree of monitors and the data on their physical appearance that I gather to evaluate the evolutionary impact of abiotic and biotic conditions. Based on the family tree and a model that incorporates plate tectonics and island emergence, I could infer that monitors originated in Africa, and from there they migrated into the Middle East, Southern Asia, then into the Philippines, New Guinea, Melanesia, and Australia. Although these results have been challenged by new fossil findings from China, which could suggest that monitors actually originated in mainland Asia. But whether they originated in Asia or in Africa, it is true that they at some point they colonized Australasia, and by Australasia I mean the Philippines, New Guinea, and Australia. So beginning in the Eocene epoch, Australia started to drift northwards until it collided with Asia. And this collision gave rise to the Philippines and to New Guinea. And it allows some groups of animals, such as pythons and elapid snakes, to colonize the Philippines, these islands in the Pacific, and New Guinea and Australia coming from mainland Asia. Arriving in a new area without competitors, these groups could fastly diversify into many different species with different habits. So imagine you are a restaurant owner and you arrive in a new town without any restaurants. So there's a huge opportunity to build a variety of restaurants, an Indian place, a Mexican place, a Thai place. So all these different, so these groups, when they arrive into Australasia, because there's no competition, they could diversify to interact with their environments in many different ways. In monitors, we see that the Philippines, Australia, and New Guinea concentrate the largest number of species, whereas other large areas, such as Africa and mainland Asia, have only a few species. So these observations seems to be consistent with this hypothesis where organisms, once they colonize the Philippines, New Guinea, and Australia, they could diversify into many different species. However, our data show that the picture is a little bit more complicated. These plots show the rates at which new species originated in the monitor groups living in different areas. So the closer they are to the right, the faster the rate at which new species were originating. And consistent with our hypothesis, the Papuan and the Philippine clade show higher rates of species origination than the monitors living elsewhere. But the rates for the Australian group, here in pink, are not much higher than elsewhere, such as mainland Africa or mainland Asia. And on the contrary, the Australian clade is more ecologically and morphologically diverse than the others. So here, for example, for body size, this stands for snout bend length, which is a measurement used to describe body size in reptiles. And you can see that there's more variation in the Australian clade here in pink than for the other clades. Both the smallest and the largest species of monitors are found in this group, in the Australian group. There's also some variation in the Papuan clade and a little bit less in the Philippine clade. In the other datasets, like here, the body shape, the shape of the head in dorsal view, and the shape of the head in lateral view. And this pattern is even more notorious. In these plots, each point represents a different species. The color represents the group that they belong to. And the closer the points are, the more similar the species are. And you can see that there's more variation, a larger area of this morphological space occupied by the Australian group than by the Papuan group here in green, and the space occupied by the clade from the Philippines, so here in yellow. Whenever I say clade, I'm just referring to a group. And the group from the Philippines has even less variation. And this pattern can also be seen in the head in dorsal view and in the head in lateral view. A lot of variation for the Australian clade, some variation for the Papuan clade, and a little bit less variation for the clade from the Philippines. So it seems like this hypothesis may be a bit oversimplistic. And instead, it appears that each group, the one from the Philippines, the one from New Guinea, and the one from Australia, may have evolved in a different way. In the Philippine group, the formation of new species was fast, and this probably has to do with it being found in islands. Islands are unstable environments, with land masses becoming isolated when sea levels rise. And this promotes the reproductive isolation of populations, promoting the formation of new species. However, there's been limited ecomorphological divergence. All these monitors look very similar to each other. So using the monitor family tree, I found that once populations became geographically isolated, they never came into contact again. Because there's no competition between the species, they are living in separate places of the world. They can all interact with their environment in the same way that they were doing it before, and new adaptations aren't necessary, explaining why all these monitors look alike. In the Papuan group, the formation of new species was also fast because they also live in islands. But contrary to the Philippine clade, there's some ecomorphological divergence. And this is because while these monitors colonized the islands surrounding New Guinea, most species still live in the central island here. And because they are coexisting with each other, they have to avoid competition by interacting with their environments in novel ways and acquiring new adaptations. If you are a restaurant owner, you won't build 10 Mexican restaurants in the same neighborhood. They will be competing with each other. Instead, you will probably open up a Thai place, an India place, a burger joint, and so on. In the Australian group, the formation of new species was slower, and this is probably because Australia is a continent, and not only that, it is the most tectonically stable continent on Earth. But they show the greatest ecomorphological diversity. This is probably because many species are all living in the same place, and they have to develop different adaptations to avoid competing with each other. But there's also other factors that could have promoted this great diversity of species and ecologies in the Australian group. Australia has a great diversity of environments. There's deserts, there's rainforests, there's rocky outcrops, there's grasslands. And monitors in Australia had to adapt to each of these very different environments, promoting a huge diversity of ecologies and physical appearances. So it seems like both abiotic factors, such as tectonics and island formation, as well as biotic factors, especially competition between different species, have shaped the diversity and physical appearance of monitors. And the diversity patterns of herpestids also support the importance of competition for the diversification of monitors. So herpestids are carnivorous diurnal mammals. An example of them is mongooses. I would prefer to say monkeys. I think it sounds cooler, but whatever. And when I was presenting this research to my colleagues at the University of Kentucky, they thought that this was the silhouette of a monitor lizard, but it's that of a mongoose. So they are very similar to each other. And because of that, and because both of them are diurnal predators, they could be competing with each other. Supporting this hypothesis, we can see that the diversity of herpestids is higher in mainland Africa, and there's also some species in mainland Asia and some of the islands here. But these mammals are virtually absent from the Philippines, New Guinea, which could be around here, and Australia. And that's where monitors are more diverse. So herpestids are probably competing with monitors in these other places, and they are winning. So that's why there are fewer monitor species living in the places where there are herpestids. And adding further support to this, a friend and I are working on a project where we are measuring different carnivorous mammals and also monitor lizards. And we see that the body proportions of these two groups are very similar to each other. So yeah, I think it's very clear that they are competing with each other. Okay, now let's talk about incumbency effects in the Australasian monitors. So we all know that coral reefs are amongst the most diverse communities on Earth, right? We have all seen Finding Nemo. And there's many different species of fish that call the coral reefs home. However, when damselfish are the first species to colonize a coral reef, the resulting communities are much less diverse, and they are instead dominated by the damselfish. And these advantages that a lineage gains by being present in a region before others are called incumbency effects. Incumbency effects have been thoroughly documented at the ecological scale, like with our damselfish example, but their importance at the macroevolutionary scale is disputed. However, there's anecdotal evidence suggesting that they are important. Here we have again elapided snakes, which arrived early in Australia from Asia, and they diversified into many different species, each interacting with their environments in different ways. There's some species that are wait and sit predators, and that look a lot like the vipers from the New World because they have a similar hunting strategy. There's some species that are semifossorial, so they live underground for most of their life. Some species are semi-aquatic, and other species climb the trees. And then we have colubrid snakes, which are the most diverse group of snakes elsewhere, but they arrived later in Australia. And they failed to diversify. There's only a few species of colubrid snake in Australia. So imagine again, if you arrive in a neighborhood that has great Mexican, Thai, and Indian restaurants, so your options for building new restaurants are going to be limited. Maybe you can open up a burger joint and a Greek place, but you cannot diversify much. And that's what happened to the colubrid snakes in Australia. So to test for incumbency effects in monitors, I took a look at the monitors that live in New Guinea and in Australia. We know that these places have been separated for long enough to have a different set of species. But we also know that they have been connected in the past when sea levels are low, providing an opportunity for the Papuan monitors to colonize Australia and the other way around. But despite their recent and repeated connection, there's only 3 members of the Papuan group that made it to Australia, and 3 members of the Australian group that made it to New Guinea. The monitor family tree also tells us that all of these colonization events are pretty recent. So I suspected that incumbency effects may be limiting immigration from one place to the other. So I fitted several evolutionary models based on the monitor family tree and found that models where the immigrants, so those monitors that came from Papua into Australia and the other way around from Australia into Papua, were failing to diversify. I also found that the immigrants are somewhat morphologically different from the incumbents that they live with. So here we have the body size of Varanus chlorostigma, which is a member of the Papuan group that colonized Australia. And this is the body size of the Australian incumbents that the Varanus chlorostigma lives with. And you can see that Varanus chlorostigma is larger than most of the species in the Australian group that it coexists with. And this pattern is the same for all the immigrants. Immigrants are either smaller or larger than the incumbents that they live with. So this suggests that only the immigrants that are somewhat different physically can survive in new places because they are not competing with the incumbents that already live there. However, these differences in physical appearance are probably meaningless because there is already a baseline difference in the physical appearance of the Australian group and the Papuan group. So if the incumbents and immigrants look different, it's not surprising because these two groups are already different. Instead, we found something that was surprising at first. We found that the immigrants are more similar to the incumbents that they live with than random species pairs taken from both groups. This suggests that it is the environment that is dictating how you look and not competition. So you have to look a certain way to be able to survive in a particular environment. And that's why the immigrants and the incumbents that live together look alike. So overall, it seems like our results are consistent with the following scenario. Immigrants arrive in a new place, like in this case, a monitor coming from Papua into Australia. And whether they can survive or not is dictated by the environmental conditions. The immigrants can coexist with the incumbents because there is a baseline difference in the physical appearance between both groups, so they are not competing that hard with each other. But however, because the incumbents are already using their habitats in many different ways, then the immigrants fail to diversify. And this makes them prone to extinction. A single species is more likely to go extinct than 10 species, which could explain why all the colonization events we observe are recent. Past immigrants may have already gone extinct and we didn't have a chance to see them. All right, now let's talk about the ontogenetic drivers of phenotypic evolution in monitors and their close relatives. And whenever I say ontogeny, ontogenetic, I mean to say growth. So ontogeny is just a fancy word for growth. As I said before, there's some variation in the body proportions of monitors. There are some species that have shorter tails and shorter limbs, others have longer limbs and longer tails. And there's huge variation in body size. Again, these are the smallest species and the largest species. And ultimately, differences between species are the result of differences in growth patterns. This is a juvenile of a salamander from the eastern US. And you can see that it has some adaptations for life on water, such as the sternal gills and the tail fin. Then it undergoes metamorphosis and it loses all these adaptations to instead live on land. But here we have the famous axolotl from central Mexico, which retains these aquatic adaptations well into adulthood. So this is a reproductive adult and it has a tail fin and has sternal gills. So it always keeps this juvenile appearance throughout its life. And I was interested in testing how these differences in growth pattern are promoting morphological variation in monitors. In this study, I also included the closest relatives of monitors, which are the Chinese crocodile lizard and the Bornean earless monitor. Okay, so these are our results, and I found that there is huge variation in how these lizards change as they grow. This plot is not as complicated as it seems, I promise. So each of these arrows represents the direction of ontogenetic change as a particular species is growing. So for example, here in the first column, if the arrow is pointing up, it means that as that species grows, the tail is becoming relatively wider. And if the arrow is pointing down, then the tail is instead getting relatively narrower. And same for the other two columns. If the arrow is pointing up, it means that the toes are getting longer as this particular species grows. And if not, if it's pointing down, it means that the foretoe is getting shorter as the species grows. Here, if the arrow is pointing up in the first column, if the arrow is pointing up, it means that the head is getting broader as the species grows, and if it's going down, it means that the species is the head of this species is becoming narrower. And for example, here in this group, you can see that there are both patterns, one in which the arrow is going up and one in which it is going down. I found that the differences between the main groups of monitors are mainly the result of differences in the direction of the arrows. So for example, in a particular group of species, they may be getting relatively longer tails as they grow, and another group of species may be getting relatively shorter tails. On the other hand, the differences between the species belonging to the same group are mainly the result of changes in the length of the arrows, and this is called heterochrony. So the most famous case of heterochrony is again the axolotl, and heterochrony results in one species. Here it would be the species in orange, showing a more juvenile-like appearance, while the other species will be more adult-like in appearance. Our results also indicate that heterochrony is responsible for the origin of the monitor body plan. So this is a young Bornean earless monitor. This is the adult of the same species, and this is an adult monitor. And you can see that the adult monitor looks more similar to the juvenile earless monitor than to the adult earless monitor. This is more evident in the relatively large eyes that both the juvenile earless monitor and the monitor possess. So this is indicating that monitors in general are more juvenile-like in a similar way to the axolotl. Heterochrony is also responsible for the physical appearance of the iconic Komodo dragon. So this is a baby dragon, and this is the adult of the Komodo dragon, and this is the adult of the closest relative of the Komodo dragon, the lace monitor. And you can see that the adult lace monitor is very similar in coloration and physical appearance to the young Komodo dragon. So the appearance of the lace monitor is the result of arrested development, while the physical appearance of the adult Komodo dragon is the result of extended development. We also found that the juveniles of the different species are more similar to each other than the adults. And we think this is because the juveniles of the different species are all living up in the trees to escape predators, including adults of the same species. The monitor lizards, most species are cannibals. While the adults of the different species are occupying their environments in different ways. Some species are terrestrial, others remain arboreal, and others are semi-aquatic. And so they have to have different adaptations for each of these different kind of habitat. All right, so we are moving into the last project that I'm going to be talking about today. And this is about the evolutionary relevance of hybridization. As you've probably heard, hybridization is the interbreeding between different species. And it has been documented at the ecological scale. We can observe it in real time. Here, the bird in the middle is a hybrid between two species of finch, each there on the left and the right. But it is unclear whether hybridization has an impact that can last more than a few generations. In other words, it is unknown whether hybridization has macroevolutionary relevance and explaining the genetic and physical differences between the species. So we use the genetic and morphological data that we gather to address this question in a group of monitors called the large Australasian monitors. I decided to focus on this group because previous studies have recovered a different family tree depending on what genes they are using to reconstruct the tree. Among other processes, hybridization is one of the most likely explanations for this discordance. And this is because evolutionary inference, the inference of family trees, can be affected when one species has the genes of another species, which is the result of hybridization. So using newly developed methods, we found that approximately 7% of the genes present in sand monitors, here in blue, which are a group of monitors living in Australia, came from the Komodo dragon, here in red. So this indicates that there was past hybridization between the Komodo dragon and an ancestor of the sand monitors. This hybridization event also had effects on the physical appearance of sand monitors. So in this plot, each point is an individual, and they are colored according to which species they belong to. And here we have the Komodo dragon in red. These are the closest relatives of the sand monitors. And over here, we have the sand monitors. And you can see that the sand monitors are intermediate in appearance between the Komodo dragon and their closest relatives, indicating that hybridization had an effect that lasted millions of years. And we can still observe that effect in the physical appearance of sand monitors. But if the Komodo dragon lives here in the Lesser Sunda Islands of Indonesia, and the sand monitors live here in Australia, then how did hybridization happen? Well, based on the monitor family tree and the distribution of the living species, we could infer the ancestral distribution of both the sand monitors and the Komodo dragon. And we found that millions of years ago, these two lineages coexisted in northern Australia. And based on the environmental preferences of the Komodo dragon and models of past climate, we also found that in the past, there was suitable habitat for the Komodo dragon, here in red, in northern Australia. However, this suitable habitat becomes restricted to the Sunda Islands of Indonesia in the present. So explaining the present-day absence of the Komodo dragon in Australia. Okay, so all of our results are consistent with the following scenario. The Komodo dragon originated in mainland Australia, likely in northern Australia, where it hybridized with an ancestor of sand monitors. Then the Komodo dragon migrated into the Sunda Islands of Indonesia, likely during a warm period where during a cold period where the sea levels are low. And you can see how close the Sunda Islands become to Australia. And then as the climate changed, there were no longer suitable conditions for the Komodo dragon to live in Australia, and then it got extinct. And this hypothesis is also supported by fossil findings. A study from 2009 found that some fossils from northeastern Australia can be assigned to the Komodo dragon. So I think it's pretty cool that genetic data, morphological data, and fossils all agree on an Australian origin for the Komodo dragon. And this has led some people to propose that Komodo dragons should be reintroduced to Australia to replace some of the native predators, such as the thylacine, that went extinct in the last 2 centuries. Our research on hybridization between the Komodo dragon and sand monitors got a lot of media attention. I'm really happy about that. It got featured in The Guardian, in Australian Geographic, also in some media in Spanish, and also in some media in Bahasa, which is the language of Indonesia. And with that, let's see what we learned today from our Scaly science teachers. We learned that both abiotic factors such as tectonics and island emergence and climatic conditions, and biotic factors such as competition between different species, and hybridization have all acted together to shape the amazing diversity of life that we can observe today. And you may have noticed that I use I and we interchangeably, and that's because this wouldn't have been possible without my amazing collaborators in Australia. These are Scott Keel, Ian Brennan with a lace monitor on his shoulder. This is Damien Esquerre with a WOMA Python, and Alex Skills. So I want to thank you all for listening. And with that, I'm happy to take any questions.
Right. Thank you, Carlos. Okay, now, Carlos, I don't believe you mentioned this, but you illustrated all of these monitors. Is that true?
Yeah. I did illustrations, yeah. And also some of the photos are mine, like the koala and the viper. And yeah.
All right, very good. So in addition to soccer and earning your PhD, you can illustrate. Something to fall back on.
Yeah.
All right. So thank you so much for presenting for TIES, which is the Teacher Institute for Evolutionary Science. And our website is tieseducation.org. We're on Twitter, we're on Facebook. And just a few months ago, we started a YouTube page where you can see this webinar and our past webinars, in addition to a ton of free resources. We have a couple of labs which we explain, but we also have short videos that you can play in your classroom. And if you go on our Facebook page, you can also see some of the past webinars. And on Facebook, I post daily scientific articles and announcements that pertain to evolution. All right, so if you want to see more exciting webinars like this, we have 1, 2, 3, 4. We have 4 more this semester, and all of our resources are free. Our labs are free, our PowerPoints are free, and if we have a lab to implement it in your classroom, it should be free, or you might have to buy like M&Ms and toothpicks, but everything is under about $5 for the full day because we know how teachers struggle with funding. Now, the only thing that we do charge for is our book on teaching evolution. And that's because we had to pay the publisher to print it. So it's a physical thing that we would be sending you. But what's exciting is today, February 1st through February 15th, we have a Valentine's Day sale. So if you love evolution, you can get the book at a discount. You can go to our website, tieseducation.org, to learn more about that discount. All right, so Carlos, we have several questions, comments. Well, we have one concern, but I'll get there in a minute. All right, okay. What do you recommend for as an undergraduate or graduate course of study to pursue studying monitor lizards or lizards in general? Or should they look for internships? How, for a student exiting high school, how can they pursue this?
Yeah, well, in my own experience, I think that most university professors and researchers are pretty happy to hear that young people are interested in science and in the research that they do. And that's the way I got into evolutionary research and research on reptiles as well. It was by approaching a teacher and telling him, like, I really like the research that you do. He was a herpetologist, and I would like to help you however I can. And then I started by putting ethanol into jars of specimens, but eventually I did my own projects.
This was in Mexico?
Yeah. But I have seen this pattern repeat itself in other places. In Australia, we had a high school student doing some research on lizards. We called him the boy genius. So yeah, I think most researchers are really happy to hear that young people are interested in this. And I think my recommendation would be to do a little bit of Googling to see who is doing the kind of research that you like. if you are interested in atoll conservation, just Google atoll conservation research. Maybe the name of a university, the one that is closest to you, and the name of a researcher working on it may pop up. And I would encourage you to email them. And I think most of them will be really happy.
That is the reoccurring theme for at least this semester. Every scientist that we've had on always says, oh, just reach out. And the scientists are just so welcoming to younger people who are interested in the same field.
Yeah, absolutely.
All right, Ryan says we have a lot of new invasive and non-native lizard species in South Florida, and they are multiplying like crazy. So this was kind of when you're talking about the incumbency. So I guess evolutionarily they're doing well, but maybe not so much on the ecosystem that they've been introduced to.
Yeah, and I do think there's a very important difference, and that's that the immigration I was talking about between Australia and New Guinea was natural and happened thousands or millions of years ago, whereas all these lizards in Florida are there because of humans.
Yeah.
So we are definitely the major factor changing ecosystems presently. And yeah, I don't know. I think we should aim to eradicate most of these species, which is unfortunate because some of them are pretty cool. Like who in Florida wouldn't like to go to their backyard and see chameleons or pythons?
Yeah.
But yeah, they are harming the ecosystem, unfortunately.
All right, so it's my understanding that with the new fossil coming out of Asia, which disrupts your initial conclusion of monitors evolving from Africa, what was your initial thoughts? Because it conflicted with your findings, right? Were you happy? Were you sad? Were you excited?
And I think I was excited. Like there are some people that are very possessive about their hypothesis, but I think that most researchers are very open to new ideas, and that's why we are into science, because it's a never evolving thing.
Yes.
And yeah, I'm happy that someone else is shedding some more new light into monitor lizard evolution.
Bertha, a 32-year veteran teacher, says, the restaurant is an excellent analogy. Great way for teachers to explore ecological niches and chances of a species to compete in an ecosystem.
Well, thank you. Yeah, I'm glad to hear that. And that's me combining my passions again. I really like to eat.
That was just one great example of just like your language of how teachers can implement evolution and make it easy to understand for students. All right, Jeko says, I like the idea of mongooses and monitors as competitors. I imagine people would first try to compare various reptiles with each other, rather than the niches. So how did you narrow it down to the mongoose as the competitor?
Well, one thing that there's a couple of things that really set monitors apart from other lizards. One of them is their size. They are larger than most other lizards. And the other one is that they are very active. Most lizards can only do a limited amount of physical activity because they rely on the environment to generate energy. But monitor lizards have a very efficient metabolism and very efficient breathing system, so they can be active for longer periods. And the size and the degree of activity that they show is more similar to that of mammals than to other lizards. So that already is hinting that probably they are competing with mammals more than with other lizards. And well, I wasn't the first person to think of this. There's been some books before that have proposed this, but they have never tested it. So I'm really excited to get this morphological data and actually test this idea.
Bertha also added, Carlos says, we, how many people are on the team and how often does he travel to Asia and Australia? I don't know if you read that or if you just happened to say it. You answered it at the very end about the team. But I know you mentioned you get to go to all these museums and measure stuff. Can you talk a little bit about, you went to school in Australia?
Yeah. I was in my PhD in Australia, and to measure as many monitors as I could, I traveled to collections in all over Australia. I think it was like 5 or 6 collections in Australia, and also some museums in other parts of the world. I went to the British Museum and the museum in Paris, the one I showed you, because they are historical collections that had people collecting all over the world for centuries. And then I also went to Bonn in Germany because there's a very active monitor researcher there. And he has, in their collection, they have some species that are not found anywhere else. Some very rare species that are only known from a single specimen. And then I also went to Hawaii because they are working on New Guinea, and they had a lot of samples from there. So yeah, I have a lot of people to thank, and as I was saying, that's why I use I and we interchangeably. And science is a collaborative effort for sure.
Which is another good takeaway for our students. Have you read any publications by Neil Shubin? Carlos?
No, I haven't.
Okay.
But I'm really interested now that
he wrote a book called Your Inner Fish, which talks about tectonic.
No, yeah. Now I know who you are talking about. Okay. I haven't read that one, but it's in my waiting list.
Okay. And then he also published a book called Some Assembly Required. And he did a great job talking about axolotls. And he talked about maybe the first like Europeans to find these axolotls. And they had them in all these tanks, and they're good for like kind of leaving alone. So they would like feed them, and then they returned months later. And there was 2 different types of animals in there. There was salamanders and axolotls. And they thought it was kind of like you putting like a dog in like a kennel, and then you come back like several weeks later and you have like a tiger and a dog. Like they were so different. And I think in the book he mentioned like Carl Linnaeus categorized them completely differently, even though they, are the same species.
Yeah. No, that sounds like a cool book. Yeah,
yes.
Definitely look it up.
All right, and then Jaco wants, this is a good question, especially for the students. How can a huge bulky species like a Komodo dragon mate with a smaller, thinner species? Because you're saying historically there was some hybridization. So can you talk about, that?
Well, yeah, that's a very good question. But we have to remember that in lizards, especially, and in some other animals as well, there is huge differences in body size, sometimes between the males and the females of the same species. So it's not that surprising. And usually it's the males of the larger species that mate with the females of the smaller species. So we think that's what happened with the Komodo dragon and the sand monitors. It was probably male Komodo dragons mating with the female sand monitors. And we also have to remember that this happened millions of years ago. So, and this happened to an ancestor of the sand monitors, not to any of the living species. So the body size could have been different, of this ancestor could have been different. Maybe they were closer in size to the Komodo dragon. But yeah, I don't think that the difference in size between a small male of a Komodo dragon and a large female of a sand monitor is large enough to prevent interbreeding.
Well, thank you so much, Carlos. The last couple of comments are about what an amazing talk. It was such an in-depth talk of evolution. And I love how multiple lines of evidence all led to the same conclusion about their migration and evolution. So thank you so much, Carlos, and thank you everyone else who attended live.
I had a lot of fun, and it's great to hear from you.